Hi, I'm André Koernig. I'm application scientist at Bruker JPK Bio AFM, and I will introduce you into the BioAFM, in the newest BioAFM, which is called NanoWizard 4 XP. AFM is a topography technique, and next to the topography, the interesting features that you can, or the interesting information that you can gain over your sample is not only topography, but also nanomechanical information. And since AFM is getting more and more, or is getting faster over time, also dynamical processes can be studied. It basically works as a finger touching over a surface, and the finger is a smaller cantilever which has tip sizes down to nanometers, and is therefore able to detect also surfaces in the sub-nanometer range or in the atomic range, therefore the name atomic force microscope. Typical samples for the BioAFM range from small molecules, peptides, proteins, DNA, over living organisms such as bacteria, cells, or even tissues. The advantage of AFM is that sample preparation is rather easy because you can measure in fluid, you don't have to do any fixation, you're not measuring in vacuum as other techniques, and still you have the atomic resolution for those samples. The BioAFM consists basically of three parts. One is the AFM head. This is where all the scanners are, the laser and the detector. Then the controller, which controls all the movement of the cantilever in the head. And of course, nowadays, the PC with the graphical unit interface that allows the user to easily control the AFM system. So this BioAFM system is now inside an acoustic enclosure and mounted on an active damping table. This is necessary as the system is very sensitive and all vibrations might disturb the measurements. One of the main features of the BioAFM is that you can mount it on an inverted optical microscope without harming the optical path. So you still can use transmission light techniques such as phase contrast or DIC, which is important for measuring on living cells, for example. This is now the AFM head, and here are all the three piezos in X, Y, and Z. There's also the laser and the detector that measures the cantilever deflection. I can just take off the head to show you where the cantilever is mounted. So this is the head. It has 3 feet, which then are used to put it on the stage, on the sample. Here you see a little transparent glass block, which is to hold the cantilever and to also let through the light of the transmission light from the microscope. The sample in this case are human lung cancer cells. The advantage of the BioAFM is that you can measure your samples in near physiological conditions. In this case, it's cells within a Petri dish heater, which maintains at 37 degrees, and you can also measure in the corresponding medium. Now we can place the head on top of these cells. And use the phase contrast of the optical microscope to position the cantilevers on top of an interesting cell. Here you can see now the transmission optical microscope. It's a phase contrast image of our lung cancer cells. And what you can see here is the cantilever which moves now over the sample and creates the image. And what you can see in this window is that you can now choose your scan region just with the optical image, and which makes it easy to choose the interesting scan region with the help of the optical microscope. Additionally to phase contrast images, you can also use fluorescence images or even high resolution optical techniques such as STAT or STORM techniques can be combined easily with the AFM and allow correlative measurements. Next to the topography, which you can see here, you get also nanomechanical information, which is correlated to the stiffness of the cells, and also Young's modulus can be extracted from these measurements. In the height image you even see, or in the topography, you can even resolve the small microvilli of these lung cancer cells. So what you see just next to here is the so-called Petri dish heater, which maintains the temperature at 37 degrees. This can be also controlled within the software. There are multiple accessories that makes the handling of biological samples easy. This is dedicated for cell measurements, but there is a huge variety of accessories which allow the better handling and maintain temperatures or optical access to your biological samples. Feel free to contact Bruker JPK Bio AFM Center if you have further questions.